day, this is Professor Will, CBMD PhD. And today's Friday, July 8th, 2022. It is 12:50 and 22 seconds, AM Eastern Standard Time. And today's research, Microbiota. So let's hop to it here. Understanding how microbiota thrive in their human host, and also told lipid production, and gut bacteria, unexpectedly, widespread. Date July 7, 2022. Source, Max Planck Gazelle Shaft. The scientists have made substantial progress in understanding how gut bacteria succeed in their human host on a molecular level. They investigated how bacteria produce inositol lipids, substances vital for many cellular processes in humans and other eukaryotes, but hitherto rarely observed in bacteria. The results indicate that inositol lipids have implications for the symbiosis between the bacteria and their host. Microorganisms like bacteria and fungi populate the human gut and contribute to many biological functions such as metabolism or immunity. In order to understand the influence of the microbiome on humans, researchers have been trying not to only identify which microorganisms are present in the human gut, but also shed light on the molecular mechanisms of the interactions between the microbiome and its human host. Researchers have now improved our understanding of these processes by investigating how gut bacteria produce a certain family of chemical compounds known as inositol lipids. Inositol lipids play important roles for many cellular processes in humans and most other non-bacterial life forms. They regulate how cells send signals throughout the body, modulate inflammation, and help ensure that proteins end up in the correct part of the larger cell. Imbalances of inositol levels are linked to diseases like polycystic ovary syndrome, the most common hormone disorder in women of reproductive age. In contrast to humans and other eukaryotes, for whom inositol lipids are essential, bacteria produce them relatively rarely. Researchers are now able to describe inositol lipid synthesis in Bacteroides, the micron, a common bacterium in the large intestine and a popular model organism in microbiome science. By developing a previously undescribed strain of Bacteroides, the micron, for which they can control whether inositol lipids were produced, the researchers discovered that these substances change the bacteria's physiology. Although we don't fully understand the precise role of inositol lipids for bacteria fitness yet, we have seen that they are necessary for a bacterium to succeed in the gut of their host. The researchers found inositol in the capsule of the bacterium, an outer layer which protects them against being engulfed by immune cells of the host. Moreover, inositol lipids change the resistance of Bacteroides, the micron, to antimicrobial peptides, substances used by the host to fight off pathogens. The researchers also described the metabolic pathways for inositol lipid synthesis in Bacteroides, the micron, that is, the chain of chemical reactions leading to the production of inositol lipids. They additionally identified a second putative pathway for bacterial synthesis of these lipids. And figuring out the pathways is interesting because it enables a prediction which other microbes might be able to make inositol lipids the same way the model organism does. And with this knowledge, researchers might even be able to engineer or manipulate inositol lipid production. The researchers look forward to future research about if and how bacterial inositol lipids might benefit the host organism. It is possible that there is a crosstalk between the bacterial lipids and the inositol lipids produced by the mammalian host. The science has come a big step closer to understanding the scale of such interactions. Good day, this is Professor Wills, MD, PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoy listening. Until next time, have a great day.